Hi everyone, so today I'm going to talk about monoclonal antibodies, MABs, in oncology. So you can just pause the video right here to scan that QR code right there so that you can get some PDF notes. These are our learning outcomes today. So what are MABs? MABs are still a type of antibody, and they are produced in labs. They are very specific and recognize and only target one type of antigen. Now we're going to talk about how MABs treats cancers. Monoclonal antibodies treats cancer by three main methods: blocking, tagging, and delivering. By binding onto a specific target on a cancer cell, so it could be like a malfunctioning receptor, an overexpressed normal receptor that lead to the abnormal growth of cells. So by blocking the target, then you think of it as like blocking the signal that cancer sends out to help it survive and grow. Without the signal, the cancer is going to die. This is a very direct action that doesn't need any other immunological support. Examples of these targeted MABs are trastuzumab that block HER2 receptor, bevacizumab that block FAG, panitumumab, and cetuximab that block EGFR. And I'm going to explain these receptors a bit more later on. While some MABs can block the signal of cancer cell, some MABs can tag the cancer, just like over here, with a big tag so that the immune system can recognize these cancer cells better and help kill them. First of all, MABs can block regulatory T cells. So regulatory T cells are what make the immune system immunosuppressive. So by removing regulatory T cells, more activated T cells can come into play and kill the cancer. Another thing that MABs can do is that they can block the signal sent from the remote control. The remote control is what we call a checkpoint, PDL1. So when PDL1 is working, it jams the signal of a T cell scanning system. When the T cell scanning system is jammed, the cancer goes under disguise and no one can recognize it. By taking off the remote control, the cancer cell can't be disguised anymore, and then the T cell will be able to locate it and recognize it. Last but not least, MABs can also put a massive tag on cancer cells so that other immune cells can come and recognize it and destroy the cancer cells. Since the first two mechanisms belong to a subgroup of MABs that is called immune checkpoint inhibitors, ICI, I won't be discussing it here today because it's a massive topic on its own and I don't have enough time to cover it. But I'll include some resources at the end so that you guys can do some self-learning yourself. And I'll be focusing on the last mechanism where MABs mark the cells for destruction. One mechanism MABs use to tag a cancer cell is called complement-dependent cytotoxicity, CDC. It is an activation of complement system. So once the antibody is attached to the cancer cell with the FAB region, the FC region will be bound with a protein called C1. C1 is the first piece of the domino. Once it's kick-started, they won't stop until it's fully activated. Once the complement system is fully activated, they will form a membrane attack complex. So this little guy here is called MAC. MAC attacks the membrane of the cancer cell. And then a lot of the extracellular fluid will go into the cell and the cancer cell will swell up and burst to death. Some examples of the CDC MABs are rituximab and daratumumab. So they are very common examples of the CDC MABs that are commonly used. The second method that MABs can tag a cancer is by antibody-dependent cellular cytotoxicity. So the MABs also bind onto the target antigen of the cancer cell, but instead of C1, this time immune effector cells bind to the FC region of the antibody. Then the activated immune effector cell use a bunch of proteins, enzymes, and cytokines that leads to the short circuit of the cancer intracellular signals. These are some ADCC MABs examples. Have you noticed that some MABs have both CDC and ADCC as their mechanisms? For example, trastuzumab, rituximab, daratumumab. Actually, a lot of them have dual mechanisms. The third tagging mechanism of a mAb is called antibody-dependent cellular phagocytosis, ADCP. Once a mAb tags onto the surface of a tumor cell, a matching phagocyte can tag onto the antibody. And then the phagocyte will eat up the cancer cell. Once the phagocyte is full, then they sort of hold up the cancer cell's limbs and legs on themselves so that they remember that they have eaten them before. So the next time they see the same cancer cell, then a more massive attack can be initiated. Other than blocking and tagging, mAbs can also deliver medication to the cancer cell. This one is super easy. So the mAbs are linked to a chemotherapy. When the antibody latch onto the target cancer cell, it then releases the chemo drug and then kills the cancer cell. The next bit I want to talk about is the naming of mAbs, which is pretty cool. So one thing everyone probably know by now, that mAb is the suffix for monoclonal antibodies. But do you know anything about the infixes? First of all, part of the infixes can tell you about the target of the mAbs, whether the mAbs target tumor, immune system, CV system, or bones. Can you think of some examples? Another part of the infix can tell you the immunogenicity of mAbs. It ranges from 100% coming from mice to 100% from human. So obviously, the closer the mAbs to human, the lower potential the mAbs is going to cause an immune reaction to human beings. It is helpful to be able to recognize the mAbs because by looking at the infixes, you know where the mAbs are targeting, so you know the potential side effects that it can bring about, as well as the likelihood of your patient having a hypersensitivity reactions from the mAbs. Now we're going to look at some side effects of mAbs. So there are some common side effects to all the mAbs because when you're using an antibody as a medication, you're always running the risk of the antibody recognizing your own body cell as a foreign antigen and attack it. That's why the common side effects of all the mAbs are very systematic, such as nausea and vomiting, hypotension, flu-like symptoms, fever, and chills. Now we're going to look at different receptors that mAb targets. Because there are so many of them, I wouldn't have enough time to talk about all the receptors today, but I'll give a few examples and I'll leave more resources at the end so that you guys can do more self-learning. So the first one is VEGF. So it is a protein that initiates and regulates the growth and permeability of vascular and lymphatic vessels. It's located in a lot of lymphatic vessels, placenta, and vascular epithelial cells. Cancer, by overexpressing VEG, they can help develop their own blood supply. Therefore, they can ensure that there are enough nutrition to feed the cancer cells. mAbs that target VEG are bevacizumab. So CI in bevacizumab means that it targets the CV system, and ZU means that it's over 90% from human. So the emphasis gives us clues that the potential side effects will be largely on the CV system and that the hypersensitivity reaction wouldn't be so high because it's over 90% from human. And now we can see that hypertension is actually one of the main side effects of bevacizumab. It is so important that before every bevacizumab administration, patients need to have their blood pressure checked. Over here, I've attached some self-learning resources. So this includes FAQ session on mAbs, so you can learn more about different mAb targets. There's a test your knowledge quiz that you can do with 10 MCQs in there. And there's the QR code for some PDF notes with extra slides in there. I've included more mAb receptors and their side effects in there as well, because I couldn't cover that within 10 minutes. And these are the resources that I've used. And thank you so much for listening.